Hello to all my dear students. So once again I welcome back on this amazing platform of Physics Wallah. Now today we are going to start the second lecture of the chapter Molecular Basis of Inheritance. Beta, in the first lecture you have just seen the beginning, the introduction portion of the chapter where we have discussed the structure of DNA. If you have still not watched that session, make sure that you first watch the first lecture and then you come to the second lecture. A continuity will be established and it will become very easy for you to understand also. Correct? So please always try to watch the lecture in sequence. It will be a good revision for you. You will be able to make proper notes. Your NCERT will be fully revised. And then yes, of course, it will be highly beneficial to you. So if you really want to take the benefit of these lectures, make sure you are watching these lectures in sequence as well as on daily basis as and when the lectures are uploaded. Now, today we are going to see one step ahead the structure of DNA, that is packaging of DNA in eukaryotes. Very, very important. In yesterday's class, I have told you about the packaging of DNA in prokaryotes, which is very simply done with the help of some positively charged polyamines in the nucleoid of the prokaryotic cell. But it is not as simple as you have seen in prokaryotes for eukaryotes. In case of eukaryote, the packaging is little bit complicated. The first step, the initial step of packaging is done with the help of positively charged proteins called histone proteins. So first point which we are going to write over here is, in case of eukaryotes, packaging, initial, basic, packaging is done with the help of, now why positively charged protein? Because DNA itself is negatively charged. So negative and positive will be attracted. So in order to reduce the thickness or reduce the length of the DNA and increase its thickness, we need some positively charged proteins around which DNA can be wrapped. And yes, it can be reduced in length. Correct? So packaging in case of eukaryotes is done with the help of positively charged protein called histone protein. Right? Now these histones are the proteins which are rich in two amino acids, lysine and arginine. So these are rich in two amino acid which is lysine and arginine. Correct? Now these two are basic amino acids. As a result, the overall charge on histone protein is positive. Now for the packaging of DNA, there are 4 different types of histones involved. There are 4 different types of histones that are involved in basic level of packaging, which are H2A, H2B, H3 and H4. Now these are 4 different types of histone proteins which are utilized for the packaging of DNA. Now see what happens. Each histone proteins in its double copy, they form a octamer. They form a spherical ball-like structure called as histone octamer. So first step in DNA packaging is formation of histone octamer. Right? So H2A की 2 copies, H2B 2 copies, 2 copies of H3 and 2 copies of H4. So this is like a positively charged ball-like structure called as histone protein or histone octamer. Right? Now this is overall it is positively charged because this octamer is made up of positively charged histone proteins. Now this positively charged histone octamer is going to attract negatively charged DNA strands. Near about 200 base pair of DNA wrap around this histone octamer in a turn. One complete turn and second turn is 3/4. So near about 200 base pairs, they wrap around this DNA in 1/1 whole 3/4 turns. So this number of DNA over here is 200 base pairs. Now just to make sure that this wrapping remains permanent and this wrapping does not again unwrap, unwinding should not take place. Therefore, to lock the structure, there is H1 histone protein. So this is H1 histone proteins which basically lock the structure, which completes one ball-like, spherical ball-like structure, histone octamer, around which 200 base pairs of DNA are wrapped in 1 whole 3/4 turn. To completely lock the structure so that the winding of DNA, wrapping of DNA does not again unwrap itself, H1 histone protein is utilized for the same purpose. Now this whole one structure is known as nucleosome. So the first step in the packaging of eukaryotic DNA is formation of nucleosome. Correct? The first step is formation of nucleosome. This is the initial level of packaging which you see in case of prokaryotes. Nucleosome, also called as new body. Nucleosome is also called as new body. It is actually made up of two components. One is histone octamer, around which near about 200 base pair wraps. So this is one histone octamer, once again see, which is made up of 4 different types of histones, each present in 2 copies. H2A, 2 copies. 2 copies of H2B. 2 copies of H3 and 2 copies of H4. So this is how the histone octamer is. It is a positively charged ball-like structure around which 200 base pairs of DNA wrap in 1 whole 3/4 turns. Again, to lock the structure, H1 histone protein is present. Now, like this, so many ball-like structures will be produced. See, this is another histone octamer. One more new body will be produced, around which again 200 base pairs of DNA. Then H1 histone proteins will ensure that these 200 base pair of DNA which has wrapped around the histone octamer does not again undergo unwinding. Now such n number of histone octamers will be produced. Now these n number of histone octamers, they will be joined to each other. See, joining is also important now, so that one complete thread-like structure is obtained. They are joined with the help of linker DNA, which is approximately of 80 base pair. So two nucleosomes, they are destined by a linker DNA which is of approximately 80 base pair. 200 base pairs of DNA, they wrap around each octamer. Such 2 octamers, they are joined with the help of linker DNA. Now, this is called as structure of nucleosome that represents the basic level of packaging in case of eukaryotes. Okay, now overall if you imagine so many ball-like structures attached with the help of string. Now, if you observe this structure under the microscope, check this. Okay, wait. Let me just add a page. Right. Now, when you observe DNA under the microscope within the nucleus, DNA is present within the nucleus now, correct? Within the nucleus, then generally what do you observe? Fleming ने क्या observe किया था? What did he observe? He observed a thread-like structure on which there are beads. This was his observation. Now basically this structure is known as bead on string structure. Beads over here are the nucleosome. And this complete string is actually the chromatin fiber. Right? The one which we usually draw, the thread-like structure made up of DNA is chromatin fiber. And when you observe chromatin fiber in the nucleus under the microscope, you see certain beads. Those beads are basically rich in DNA called as nucleosome. So bead on string means nucleosome arranged on a string-like manner. That string is basically chromatin fiber, which during the time of cell division condenses to form chromosome. Now, these are the two only winding and unwinding, decondensation and condensation. If genetic material has to decondense, it will only decondense to form chromatin fiber. It's not like during interphase, the whole DNA nucleosome will also unwind. No. Never ever nucleosome unwinds. Nucleosome remain in that condensed form. It's simply chromatin fiber in interphase, and during dividing phase that chromatin fiber further condenses to form rod-like chromosomes. Right? So the basic level of packaging is done with the help of histone proteins, which is making nucleosome and then leading to the formation of bead on string structure. Now when this bead on string structure further condenses to form thick rod-like structure during the time of cell division, to form thick rod-like chromosomes. Right? Basic level of packaging is done with the help of histone protein, but this is higher level of packaging. This conversion of bead on string to chromatid or chromosome is higher level of packaging, which is done with the help of non-histone proteins. So higher level of packaging in eukaryotes is also done with the help of non-histone proteins, but basic level of packaging, that is conversion of DNA into nucleosome, is done with the help of histone proteins. 4 different histone proteins are involved: H2A, H2B, H3, H4. H1 is used to lock one structure of nucleosome. I hope you all have understood whatever information is right now given on the board. So this you have to do in packaging of DNA under eukaryote. Right? Chalo. Now you have seen that, in the beginning of the chapter only we declared that DNA is the genetic material which makes up the gene, and then these genes, as when required, they produce protein and regulate the metabolic activity of the body. You have already seen the structure of DNA, the importance of DNA, who proposed this structure, Watson, Crick, everything you have seen, central dogma, reverse. That means you have realized so far that DNA is actually the main nucleic acid that acts as a genetic material. That's why ma'am is teaching you the structure of DNA but not RNA. But there must be a time like where scientists were maybe not knowing that whether DNA is the genetic material, RNA is the genetic material, or protein itself is the genetic material. There must be the time. So now we are going to that era where people were not aware about what is genetic material, and scientists were performing sets of experiment to prove that DNA is the genetic material. So now the next heading is search of genetic material. In this, the very important and the first experiment, recognizable experiment, was done by Griffith. Griffith, while working on bacteria Streptococcus pneumoniae and mice, tried to show that DNA can be the genetic material. However, he could not prove this. I'll tell you how when we will proceed with the experiment. Chalo. So now we are going to learn about the experiment by Griffith. He was working on bacteria Streptococcus. Okay, so I'm sorry for the disturbance. I don't know how my nib just got broke. I hope everything is fine. Chalo, now let's just see. It must have fallen down and I was completely unaware about it. Okay, so now we are talking about the bacteria which is Streptococcus pneumoniae. Now, this Streptococcus pneumoniae beta bacteria, there can be two types of Streptococcus pneumoniae. One S3 strain and another R2 strain. S3 strain, S stands for beta smooth or virulent bacteria. Smooth or virulent. Such bacteria whose outermost layer is present in the form of thick, tough, resistant capsule. Then such bacteria, if they enter in your immune system, if they enter inside your body, then the host immune system fails to recognize them, and then they multiply in the host body, and then they cause pneumonia. Then they can cause disease in the body. So if S3 strain of Streptococcus pneumonia enters in a human body, it will be very easily able to cause pneumonia. Correct? So smooth is basically the virulent strain whose outermost layer is called as capsule, which is thick, tough and resistant layer. Hence, they are not recognized by the immune system and they cause disease in the host body. On the other hand is R2 strain. R stands for rough or non-virulent. In such bacteria beta, the outermost layer is not capsule. Rather, it is some slimy layer called slime sheath. They don't have capsule, rather they have slime layer. So when capsule is absent, such bacteria, if they enter inside your body, host immune system will recognize them, will kill them, and they will not be able to cause the infection. Now, understood? So basically, Griffith took two strains of pneumonia strain. One was disease causing, another was non-disease causing. Now, parallelly, he took a mice. Okay, this is my mice. Now, to first mice which he took, he injected the Streptococcus pneumoniae bacteria of R2 strain. Now tell me, if this mice is injected with R2 strain, will this mice die or not? Of course, it is not going to die because R2 strain is non-virulent. This R2 strain will not cause any infection in the body of the mice and mice will live long. On the other hand, if the same mice is then injected with S3 strain, what will happen? the mice will die. Why? Because now the mice will develop the pneumonia. Griffith did not give any medicine to cure the pneumonia. Hence, mice died. I hope first two cases clear. In the third case, what he did, he first heat killed this S3 strain. Heat killed means first he exposed the bacterial strain S3 in a heat. Heat diya. Heat beta, when heat means high temperature. So when you expose bacteria to the high temperature, automatically at very high temperature, the proteins, they denature. Now capsule is a mucopeptide layer. Basically it is made up of protein. So when we give high temperature to S3 strain, the capsule gets denatured. If capsule gets denatured, then automatically S3 will start behaving as R2 and mice will be alive. So now when over here heat killed S3 strain was given to the mice, mice was alive. And what was the simple reason associated with it? The simple reason associated with it is heat killed S3. They denature the capsule, they lack capsule, hence they start behaving as R2. Correct? I hope first three conditions are clear. Now, moving towards the fourth condition, which is very, very important to understand. Now this is one more mice. Now to this mice, an injection was given. First of all, before giving an injection, Griffith prepared a medium of R2 strain along with heat killed S3 strain. So first beta, what this fellow did, he prepared a medium in which he cultured both the bacterias, R2 bacteria and heat killed S3 bacteria, and then injected in the mice. To his surprise, the mice died. Now see, when R2 was given alone, mice was alive. When heat killed S3 was given to the mice, still mice was alive. So what happened when both the bacterial strains were brought together, when they both were cultured together and the culture injection was given to the mice, then what happened that actually the mice died? Griffith got confused that, array, when R2 given alone, mice was alive. When heat killed S3 given alone, mice was alive. So now what happened? What happened actually between R2 and heat killed S3? That together they killed the mice. So for this he concluded his experiment by proving that heat killed S3, see, they don't have protein. Now heat killed S3, they don't have capsule, because capsule got denatured due to the exposure of high temperature. Now this heat killed S3 is itself behaving as R2. So alone it cannot kill the mice. But this heat killed S3 is having the genetic material, which is capable of synthesizing the same capsule protein again. Correct? It's just like you go to the barber shop and you ask him to make you bald. So you come back home, your mummy gets surprised to see you bald. But then what do you say? That mummy, don't be surprised. It's only external hair has gone, na? But inside the proteins which are responsible for producing new hair, they are still there in my body. 1 month later, see, all my hair will come back again. So same is the case over here also. Heat killed S3 strain, you have just denatured the capsule. You have just removed the outer proteinaceous cover. But you have not removed the genetic material which is responsible for synthesizing the same protein again. Correct? So what happens now? This heat killed S3 strain releases its genetic material in the medium in which you have cultured the bacteria. Now this genetic material beta is capable of synthesizing capsule again. Right? Have you all understood? Is capable of synthesizing capsule again. Now this released genetic material is then picked up by the R2 bacteria, which is also cultured in the same medium. Now that infectious genetic material has passed from S3 to R2 through the medium. Now R2 will pick up the genetic material and it will synthesize the protein. That protein will produce capsule around R2, because of which this R2 will start behaving as S3. As a result, mice will develop the disease and will die. This simple experiment is called as transformation, because over here you see transmission of genetic material from one bacteria to another bacteria through a medium. See, R2 and S3, they are not coming in direct contact. It is only through the medium exchange of genetic material is taking place. This is known as transformation. So this whole experiment by Griffith beta is known as transformation. What is transformation? Just check on the board. Meanwhile, I am writing the definition. Exchange of genetic material between the two bacteria through a medium without coming in contact with each other. Clear? Have you all understood that how the fourth mice died when he got injected with an injection having the culture of R2 and heat killed S3 strain? If you have understood it so far, then now let's proceed further. See, this is the experiment by Griffith. Griffith's aim was simply to show the exchange of genetic material. However, he could not prove the nature of genetic material transferred from S3 to R2. He simply in his experiment, he simply in his conclusion mentioned that okay, there is some genetic material which is getting transferred, but he could not decode the nature of genetic material. He could not decode the nature, that whether it is DNA, RNA or protein. So later on, the same experiment was repeated by 3 different scientists to prove that DNA is the genetic material that is being transferred from heat-killed S3 to R2, and R2 by gaining that DNA makes the capsule and again becomes virulent. Clear? So now we are going to check out the experiment by Griffith was further continued. This experiment was further continued by 3 scientists. Who were those 3 people who continued the experiment, beta? Avery, MacLeod and McCarty. These 3 people, once again I am repeating the name, Avery, MacLeod and McCarty. Let me write it properly. Now my nib has just broken down, so I am not able to write it well. Avery, MacLeod and McCarty. These three scientists continued the experiment by Griffith, and they proved that DNA is the nature of the genetic material which is getting transferred. Now see how. Beta, they took 3 test tubes, right? They took how many test tubes? 3 test tubes. In each test tube, they prepared the same medium containing heat-killed S3 along with R2 bacteria. The same. In all the 3 beakers, in all the 3 test tubes, these 3 people added the same medium having heat-killed S3 and R2. Now, in first, they added, along with the medium, they added the enzyme DNase. In second, they added the enzyme RNase. And in third, they added the enzyme protease. Clear? Now, one by one, they injected the injection of all the medium to mice. So this is A mice, this is B, and this is C. A mice is going to get the injection from A test tube. Now, when he gets the injection from A test tube, surprisingly, the mice was alive, did not develop the disease. Remaining both the mice, they received the injection from B and C test tube, and they both died. Now, can you conclude that why the A mice is the only one which is alive and why B and C could not survive when they get the injection from B and C test tube? Can you? If not, then listen to me carefully. Now, beta, if A mice who's injected with the injection from A test tube is alive, this proves that DNA was the genetic material that got transferred from heat-killed S3 to R2. How? Now beta, this heat-killed S3, what it will do in the medium? It will release its genetic material, and then that genetic material will be picked up by R2. Now, if that genetic material is DNA, as soon as DNA will come in the medium, DNase enzyme is present. So this DNase will break the DNA and R2 will not get any genetic material. R2 did not pick up the genetic material. R2 remained as R2. It did not become virulent. So mice is alive. But in case of B, enzyme RNase is added, which is only capable of degrading RNA but not DNA. Correct? So heat-killed S3 is actually releasing DNA. RNase will not do anything with DNA. R2 is free to accept this DNA. It will get converted into S3 and mice will die. Same thing happened with the C test tube also. Now DNA was the genetic material which was released by heat-killed S3. This DNA has no effect of protease. Protease enzyme can only degrade protein but not RNA or DNA. So DNA will remain intact. It will remain unaffected in the medium. R2 will get the opportunity to pick up that DNA from the medium. With the help of that DNA, it will synthesize protein. It will make the capsule around it. And now it will change the nature from non-virulent to virulent. As a result, mice will develop the disease and hence it will die. So since first test tube mice is alive, that proves DNA is the genetic material because R2 could not gain any material from the medium. Correct? It remained non-virulent, so mice did not develop any disease. Like this, with the help of these three scientists who continued the experiment of transformation, who continued the experiment by Griffith, proved that DNA is the genetic material but not RNA or protein. However, later on, two more people came by the name Hershey and Chase. They 100% proved. See, every MacLeod McCarthy, 99% they said that yes, DNA can only be the genetic material with the help of the R experiment. But it was some scientists still were like who did not believe in them. For them, two more scientists came by the name Hershey and Chase, and they finally gave the unequivocal proof. Unequivocal means the proof in which there is no question mark. Nobody will raise any question that no, your experiment is wrong, okay, why this, why that. Unequivocal means 100% truth. So 100% truth experiment was performed by Hershey and Chase to prove that yes, DNA is the only genetic material. These two people, they worked on bacteria Salmonella typhi, right? And they worked on bacteria E. coli and virus bacteriophage. Just change that Salmonella typhi was not the bacteria on which these two people worked. You can just mention E. coli, and they used the virus bacteriophage. Correct? So remove that Salmonella typhi from there, and instead of that write virus bacteriophage. You all know what is bacteriophage. It is a virus that attacks bacteria and has the potency to kill it. Structurally, it is resembled by a head, a polygonal head, followed by collar, neck, tail, and 6 tail plates, 3 on each side. The genetic material is DNA, that too double-stranded DNA. This is how bacteriophage looks like. Now, what Hershey and Chase did? Beta, they chose two bacteriophage. In first bacteriophage, suppose this is a bacteriophage. First bacteriophage, DNA, okay. Wait, let me tell you one more thing, beta. Viruses are simply nucleoproteinaceous structure. They only have two components in their body. Nucleic acid, which over here is DNA, and rest all body, the head, tail, everything what you're seeing is only made up of protein. So chemically, viruses are only having two components, protein and nucleic acid. Proteins have sulfur in them, whereas DNA has phosphorus in them. So Hershey and Chase, what they did, they took two bacteriophage. In first bacteriophage, they labeled their genetic material with the help of a radioactive substance P32. So the DNA is now labeled. You know, doping, kind of doped. The genetic material of a bacteriophage is doped, is labeled with the help of radioactive P32. Correct? Now they took one more bacteriophage. Whose DNA was normal, non-radioactive, but their protein, that is their body, was labeled, was doped with radioactive S35. Radioactive S35. Now, how many bacteriophages do we have? We have two bacteriophages. One whose, capsid, whose body is labeled with S35, having protein. And in second, the DNA is labeled with radioactive P32. Now, this is the first condition. Now, first setup, this is just the, this is the setup of the experiment. Now let's start the experiment. For this you also need two bacterias. So now these are two E. coli bacteria, unicellular organisms having their genetic material which is nucleoid. Now these are my two E. coli. So now requirement is ready. Requirement of the experiment is to have two E. coli bacteria and two bacteriophages, in which one is labeled with P32 and another is labeled with S35. Now the first step, let's start the experiment. First step of the experiment is infection. Infection means let the bacteriophage attach with the E. coli. Infect the E. coli bacteria with the bacteriophages. So now both the bacteriophages will infect. And suppose this is A. E. coli, this is B. E. coli. So A. E. coli is having P32, whereas B, this is B. B bacteriophage, which is going to infect the B. E. coli, is having S35. So first step, infection, is done. Now second step is agitating, that is blending. Agitating the mixture. Now beta, what happens as soon as infection step is done? As soon as you allow bacteriophage virus to attach on the body of bacteria, what happens then? This head of the bacteriophage along with the genetic material, they also have certain enzymes like lysozyme. That lysozyme soon after infection punctures the wall of the bacteria and lets its genetic material enter inside. I am repeating once again, listen to me carefully. What happens beta? These bacteriophages head, apart from having genetic material, they also have enzymes like lysozyme. As soon as infection step is done, as soon as bacteriophage attaches to the E. coli, now the lysozyme enzyme is being released, which punctures the wall of the bacteria. As a result, only genetic material, no head, no neck, no tail, nothing. Only genetic material, which is double-stranded DNA, enters inside the bacteria, leaving the whole body outside. You can simply say DNA comes inside and proteinaceous body remains outside. This is infection. Now second is agitating the mixture so that the DNA has entered inside. Now whatever body is attached on the bacteria, it should get separated out. So to separate the remaining portion of the virus from the bacteria, we have to perform blending. So now what happens during blending? So this is the condition. The two E. coli bacteria, which are also having now the genetic material of virus along with their own genetic material. After blending, the remaining body of the virus will get separated. The virus will get separated and genetic material carrying E. coli will also get separated. Now beta, they are cultured in a medium now. They are there in a medium. So it's like this. Now, the body of the virus is separated. Now, the third step is centrifugation. Centrifugation means in a medium of cesium chloride, you are going to measure the density of the E. coli and the supernatant. Supernatant is basically the medium. So now this is my bacteria, right? And this is my remaining medium. So now with the help of centrifugation, in the medium, basically centrifugation is separation of two components on the basis of density in a medium having cesium chloride. So centrifugation means separation on the basis of density in a medium containing cesium chloride. Correct? Now see beta, what happens over here? Soon after centrifugation, the density, now this is the A result. A medium result, I'm going to write over here, and here I'm going to write the B result. Right? Now what happens in case of A result? The medium, which is called as supernatant, which is having the body, the virus body, it will be non-radioactive, it will be lesser heavy. Correct? The A1. Whereas the bacteria of density will be more. So in case of A, inside the test tube having cesium chloride, the bacteria will settle at the base because their density is more, and the medium, the supernatant, will be placed above it. The reason is that the density of supernatant is less and bacteria is more. In B case, it will be just the opposite. The supernatant will be heavy and bacteria will be lighter in weight. Now what is the reason? The reason is radioactivity. In case of A, the radioactivity enters inside the bacteria along with the phosphorus, because you know in case of A, we have labeled the genetic material with radioactive phosphorus. Now since DNA is the genetic material that enters inside the bacteria, it carries the radioactivity inside the bacteria. Simply the virus body, which is non-radioactive, remains in the supernatant. So supernatant is light, but where radioactivity is present, that portion becomes heavy. Now, what happened in the B case? In case of B, the radioactivity was present in the body, which is protein, which remains in the supernatant, and genetic material, which is DNA, is free from radioactivity that enters inside the bacteria. Right? So bacteria becomes lighter in weight, but supernatant having the radioactivity becomes heavier in weight, so it settles down at the in the test tube after centrifugation. Had it been not DNA the genetic material, if protein would have been the genetic material, then the condition would have been reversed. Then in that case, A would have had lighter bacteria, whereas B would have had heavier bacteria. So like this, since DNA is only the genetic material but not protein, so result is in front of you. DNA entered inside the bacteria, which is the genetic material, carrying the radioactivity, making the A bacteria heavy, making it settle down at the base of the test tube. So, with this simple experiment of Hershey and Chase, we came to know that DNA is the only genetic material. To support them, already every MacLeod McCarthy did the experiment, which also had the same conclusion of DNA being the genetic material. Like this, the search was done and DNA was universally declared as the main genetic material. However, some viruses like tobacco mosaic virus, HIV virus, in some viruses you see that RNA can also act as a genetic material. But in general, 95% of the cases except viruses, all organisms are having DNA as the genetic material. So over here, our search is also done. Now we are proceeding with the next topic, a very simple topic which is going to help you decide that why DNA, why not RNA or protein, why Griffith, Avery, MacLeod, McCarthy, Hershey, Chase, all their experiment were showing that DNA is the genetic material, but not why RNA, why not protein. So beta, there is like certain there are certain properties which a genetic material should follow. If any genetic material is not following that property, it cannot be considered as a genetic material. So here is the list of the properties which a genetic material, that is DNA or RNA, should exhibit. If they don't exhibit, they will not be considered as genetic material. And now let's check that why DNA won the race over RNA. First is, it should be able to generate its replica. Means if genetic material has to be a genetic material, then it should be capable of producing its own copies. So you know, DNA can directly undergo replication and RNA can also undergo replication via DNA. Yesterday I told you reverse central dogma. In case of some viruses, RNA is the main genetic material. From RNA, using RNA as a template, we first obtain DNA and then the DNA can make its own copies. So first point is correct for both DNA as well as RNA. Second, they should be stable. Now, stability was the only factor which RNA could not pass. RNA, due to the presence of uracil in place of thymine and due to the presence of hydroxyl group at their 2' position, these two reasons basically remove RNA from the league of becoming genetic material. RNA is highly catalytic, it is highly unstable, it can undergo mutation very fast. Mutation should take place. This is one of the property of genetic material. Slow mutation should take place. But RNA, due to the presence of free hydroxyl group, due to the presence of uracil in place of thymine, they undergo mutation at a very fast rate. They are catalytic, they are very reactive, they are not stable. And if any chemical is not stable, it cannot be considered as a genetic material. And because of this reason only, RNA was removed from the race and DNA won the race and was considered as the main genetic material. Correct? Understood? Third, it should provide the scope for slow changes. I have already told you DNA can undergo mutation but not as fast as RNA. Very fast mutation is not required. Mutation should take place but at a slower rate. Clear? Fourth, it should be able to express itself in the form of Mendelian characters, which are genes. So genes are chemically made up of DNA. DNA for making protein has to convert into RNA. So you can simply say that chromatin fibers, correct, in which genes are present, they consist of both DNA and RNA. So both DNA and RNA can express themselves in the form of Mendelian characters. Only where DNA won and RNA lost the match is stability factor. Because of which you see all the scientists who performed the experiment, they ultimately came to the conclusion that instead of RNA, DNA is the genetic material. However, I told you in some viruses, RNA can also behave as genetic material. With this, we come to the end of today's lecture. Today we have done a lot many things. You have seen the packaging of DNA in eukaryotes, plus you have seen the search for genetic material and the properties of DNA versus RNA for becoming genetic material. I hope all these simple short topics are clear to each and every one. All the experiments are clear to each and every one. If yes, then these are certain questions which you have to do. I hope you all are going to do these questions and watch the lecture sincerely. Till then, thank you. Bye-bye. Take care.